still remember the first time I've learned about the axolotl. This small salamander can regrow its limbs, its spine, and even its brain. We can literally take big chunks of its brain out, and it will fully regenerate. Unfortunately, humans do not share this superpower, at least not yet. But today, we're going to talk about how we're going to take this superpower back. Now, in 1913, the father of modern neuroscience, Santiago Ramon y Cajal, have stated that the neuronal paths are something fixed and immutable. Everything may die, nothing may be regenerated. This statement really shaped our field forever. However, Cajal also stated something less quoted. This is the duty of the next generation to overcome this intrinsic failure of the brain to regenerate. And here we are today, not quite next generation, more like 4 or 5, but trying to tackle Cajal's challenge. My lab accepted Cajal's challenge. During development, we gain most of our brain mass through 2 main processes, gliogenesis and neurogenesis. Gliogenesis, stem cells divide and differentiated into glia, once dismissed as glue. Today we know that this cell type is super important. It mediates inflammation, circuit formation, and more. And neurogenesis, stem cell divide, proliferate, and differentiate into neurons, the most famous cell types in our brain. These are the cables in our brain. They transmit signals. They mediate everything you're feeling now. And here is a key point. Very early after birth, this process is almost completely shut down, which basically means that you're going to have to live with all of your neurons all throughout your life. Which means if neurons are dying, there is no coming back. And so there are multiple brain disorders and conditions that killing neurons. Aging, Alzheimer's disease, Huntington's disease, and more. Each affect different brain region. Each has a different phenotype. But one common phenotype is neuronal loss. Most treatment today aiming to stop or slow disease progression. What we are trying to do is to reverse disease progression. Now, when you think about it, there are 2 ways to go. First is engraftment. We could take cells and inject them directly into the diseased brain regions. Another approach is recruitment. We could target cells that are already within the brain and convert them into neurons. Now, both approaches are exciting, and in my lab, we're working on both. But today, I'm going to focus about the recruitment part, because I believe this is the new frontier of brain regeneration. Now, if you would ask the scientific community, can you really change cell identity? Only 20 years ago, you're going to get the answer, no way. Scientists will show you this Waddington's developmental landscape and tell you that stem cells begins at the top and then slowly rolling down and have to find the right path towards maturation. They can become glia, neurons, and other type of cells in the brain. However, science is super cool. And today we know that that's not true. Cells can be nudged, redirected, transdifferentiate. Mature cells can jump between states. We can also go all the way back into stem cells state, which to me really open avenue for therapy. But it also raising pretty practical question. If we can redirect cells, where should we start? As a parent, I've learned that changing doesn't go with force, but with readiness. It looks like cells are the same. I would argue that the best place to start would be within these dormant neural stem cells. Now, what do I mean? What is dormant neural stem cells? As I told you, during development, stem cells can become glia, neurons, and other type of cells, and they're active. But very early, these stem cells become quiet, dormant. They're still there, sitting in the two neurogenic niches, the subventricular zone and the subgranular zone. Quiet, but they are there, waiting for the right cues. What if we could target these cells and unleash their potential to become neurons? In my lab, standing on giant shoulders, we developed a framework to study neurostem cells. We define neurostem cells, we modulate neurostem cells, and we enhance neurostem cell-based therapy. For me, defining neurostem cells is the hardest part. But today, thanks to the development in technology, we have a really good shot. Think about it. If before you were looking in satellite map, you will only see lights. Today, we could dive in and see the roads. We could see the cars and even who is driving. Similarly, in cell biology and neurocell biology, if before we could see only bulk, today we could dive in and see even intracellularly. What do I mean? Here you go. What you see here is a cross-section of the 8-week-old mouse brain. Each dot represents single cell that we were imaged with our spatial transcriptomics technology. I'll spare the details, but basically what we can do is quantify different RNAs within single cells. Cells with similar RNAs going to have the same color, the same identity. And voila, the architecture of the brain appears. Pretty cool, right? Now, what about neurostem cells? So in order to study neurostem cells, we went back in time and imaged the developing mouse brain. And then we dived in. What you could see here in yellow are the neurostem cells. In blue and pink, these are the immature neurons that were generated from their activation. You could see that they are located in the two neurogenic niches and that this process is super active at this stage. When we perform the same assay in the 1-year-old mouse brain, we see that neurogenesis is almost not there. This process is almost shut down. But we can still quantify the neurostem cells here in yellow, as you could see in the two neurogenic niches. Quiet, but they are there. And here is the question that really keeps me awake at night. Why? Why these stem cells, if they are there, are not active? Think about it. If you injure your skin will fully regrow, regenerate. However, in the brain, this is not the case. So how could it be that the most important organ in our body cannot regenerate? How could it be? We don't really know the answer, but we have clues. One theory is that the brain is much more complex, so it's much more complex to regenerate. Another theory that I like even more is that the brain plays it safe. Uncontrolled growth in the brain could be detrimental, can cause epilepsy and modify function. So the brain locks off. And so the trade-off might be locking off versus regeneration. We cannot get regeneration, but we are safe. If we could unlock this blockage, we might be able to generate new neurons in the brain. Now, how are we going to do that? Let's go 1 second to our skin example. Every cell in our body, including skin, glia, and neurons, have the exact same DNA. So it's not about the DNA, but it's about RNAs. The same RNAs we are quantifying with our spatial transcriptomics. These RNAs are actually the molecular brakes. So if we could play with them, we might be able to change identity. Now, comparing the developing brain with the 1-year-old brain, as I showed you before, we found one of these breaks. Now, how can we change it? How can we downregulate it? What we did, and one very attractive approach, is using antisense oligonucleotides. These are small DNA drugs that allow us to target these specific breaks and removing them. So at this point, we have this DNA drug. We deliver them all throughout the CNS or the central nerve system and remove this break with the hope of unleashing neurostem cell potential to become neurons. And that's what happened. What you could see here is just a control one-year-old group. You see that there are almost no new neurons. However, 2 weeks post-injection of the ASOs, there you go. What you could see is the reappearance of the new immature or baby neurons, here in pink or in green. We unleash neurostem cell to differentiate and become new neurons, new immature neurons. At this point, you might ask, how do these neurons know where to go? So we don't do much at that point. We're just relying on biology. Similarly to baby turtles, when they're born, they know where to go. Even if the ocean is kilometers away, our new baby neurons know where to connect. They send their hands, dendrites and axons, to the right place. You could see that they mature here in red and connect to the right place. They fire action potential. They're functional. Most surprisingly, they could even modify mouse behavior. So when we tested a group of mice that were injected with these DNA drugs, we see that they find the way out of the maze faster than the control group. So these neurons are actually meaningful. So what I've showed you thus far is that a single injection of a DNA drug into the mouse brain can unleash neurostem cells from their dormancy, and we can see the appearance of new immature neurons that mature and behave. We literally engineered the brain, the mammalian brain, by a single injection of these DNA drugs. Next step is efficiency, timing, durability, and safety. But at this point, we have to stop. Before everyone taking their phone out and calling their grandmas, we have to mention that everything I showed you thus far is in the mouse brain. And mouse are not human. The human brain is much larger, much more complex. Every scientist will tell you that studying humans is much harder. And this really brings me to one of the most active debates in the neuroscience field today. Different groups from both sides were arguing whether new immature neurons can naturally appear in the human brain. Some groups suggested that they saw bona fide generation of new neurons naively in the aging brain. Other groups says that they couldn't find it. They couldn't recapitulate this. And that's the moment when it hits me. While scientifically important, I think that measuring new immature neurons in the naive aging brain won't cure brain diseases. I suggest we refocus. I call it therapeutic neurogenesis. Rather than looking for the immature neurons naively, let's focus on the dormant neurostem cells. Let's try to find them, control them, and enhance neurostem cell-based therapy. The implications are huge. Imagine an ALS patient start walking again, and Huntington's disease patient where his tremors stops. And the implication goes even farther. It suggests that the brain is not a machine with broken parts, but it's a machine with different states. Then therapies should target these states. Now, at this point, I have to say I'm not alone in this effort. There are top-tier groups around the globe trying to unleash the potential of the dormant neurostem cells to become neurons. But we need much more attention, funding, and resources. How are we going to get there? Only together. I think that both scientists from different approaches, engineers, and you, us, should sit all together and try to ask the right question and in a targeted way. Can we actually generate new neurons today in the human brain? No, not yet. But I believe that this is the duty of our generation. We can do it together. If our parents thought that HIV is uncurable, today they know it's wrong. We start at neurodegeneration. We can prove Cajal was wrong, together. History does not repeat itself, but it rhymes. Thank you so much.